So what I'd like to talk to you about today is cost-effective trio sequencing on AVITI. So nice to talk about the technology, but I really love to talk about the applications because that's the stuff that really matters. This is a wonderful collaboration that we've had with UCSD and some of our ecosystem partners. For those of you unfamiliar with rare disease, it's really a huge problem. Disease is considered rare if it occurs in fewer than 1 in 2,000 people, but collectively there are over 7,000 of these rare diseases, so collectively tens of millions of people are affected. And the diseases tend to be very severe. They affect disproportionately children. They're responsible for a large fraction of pediatric admissions and long-term admissions. And because any one of the diseases in itself is rare, when you go to your local doctor, they have no idea what to make of it. Extremely difficult to make a diagnosis, and people get stuck in this incredibly painful diagnostic odyssey that lasts years, procedures, misdiagnoses. So as a sequencing company, what is our role here? And of course, genome sequencing for rare disease has been done with great success, but I'll give you our unique spin on it. So this collaboration in particular is with Dr. Radha Ayagari and her student Pooja Biswas, and they study retinitis pigmentosa, so it's early onset blindness. And what we did in the past, and if you saw our launch event, I presented those results, we were able to solve about half of the cases that we were presented with. And this time we decided to start with a brand new case that we had never seen before, but to sequence the trio. So that is to sequence the child as well as the two parents. Their DNA samples were also available. Now, why would you want to sequence a trio, first of all? Well, sequencing the trio enables you to confidently call de novo variants, so ones that arose in the child, did not exist in the parent. That's how you might explain the fact that the parents are healthy, but the child is not. It can also help you to verify a compound heterozygote and trace the inheritance pattern of your mutation of interest. Now, the AVITI specification is 800 million reads, and if you do the math, that means that, it's unclear if you could really fit 3 genomes onto a single flow cell. Our sweet spot sort of 2 whole genomes per flow cell. But then we thought about it and we said, well, we could get a coverage of, let's say, close to 20x for each of the parents and then upwards of 40x for the child. Is that going to be sufficient for what we need to do? So then we started working with the Google team, and they have a tool called DeepTrio, and synergistically on one phone call, their lead developer said, oh, we actually don't need a lot of coverage in the parents, and it still works really great for de novo discovery. And so, oh, wow, okay, let's test that out. So the way we designed this, since we have the indexing, we could just do twice as much sequencing for the child, half for the parents, and see what happens. The implication of this is that now the entire trio costs $1,680 in reagents on a benchtop instrument, which I think is pretty unprecedented. So now, of course, the big question is whether it works. We have our ecosystem of partners, and this is just illustrative. We have many other partners in these spaces, so you can interchange components depending on the technologies you like to use. But what I would like to highlight here is Roche library prep with our Elevate kit, the AVITI sequencing, of course. Here we used Google DeepTrio for the reasons that I previously outlined, and then Genoox for interpretation. Let me tell you a little bit about our informatics partners here. But first, the Upstream Prep. So this is the Element Elevate library prep, which you can use. You can start with DNA or RNA. You pick one of the many supported third-party kits. In this case, Roche. You generate this linear library, which is circularized, put on the AVITI, and then a standard FASTQ file comes out. This particular prep is compatible with many third parties. It allows for the multiplexing that we talked about, 96 a day, to be extended later. Very, very highly repeatable, and comes with accurate quantification to address the question that was asked before about how we control the number of features on the flow cell. So that is the library prep. Many people more qualified in this room to talk about that portion of it. So if you have follow-up questions, I can point you to the right person about the details. So let me tell you just a couple words about DeepTrio. Here's their publication. You can read more details, but the essence is that they jointly consider data from the parents and the proband, and that improves their overall accuracy, and in particular improves their ability to call de novos with high confidence. Andrew Carroll from the Deep Variant team will be presenting as part of our workshop. He'll be talking about Deep Variant more generally, so I encourage you to attend that on Wednesday if you have the time. One company that we also really like to use is Genoox for interpretation. So they have a community-based approach. You can easily connect with other researchers studying the same disease that you're interested in. They have a very beautiful user interface for interpretation. And it's actually completely free to use until you want to customize and sign out reports. That level of integration, then they start to charge for. But for the type of application that we do here, it's just a really great fit. They are in many of the leading institutions. You can see on the bottom left display. So the screenshots that I'll be showing in the couple of slides to come all use Genoox. So we've done one run, so one trio, just as kind of an announcement. We'd love to extend the study further. So for those of you interested in rare disease, and especially those of you who have samples, come partner with us. ASHG is going to be here in no time, and we'd love to have a much more comprehensive version of this talk available for that venue. But let me tell you about the one run that we did do. You can see that the specification of 800 million reads was surpassed by a lot here. We had closer to 1.1 billion reads of 2x150. The Q30 is still in the order of 95%, a very nice high assignment rate that gave us 326 G of output. And our coverage, we were counting on 19, 19, 38. Instead, we got 20, 22, and 52. So this was a really good starting point for the analysis. You can see in the display over here, this is the indices corresponding to the proband, the indices corresponding to the parents, and then our phi-X controls there. As you can see that indexing worked really well and gave us the balance that we wanted. Another quick look at summary metrics before I get into the biology of the case. You can see we report a mean quality score. You can see that it starts and stays above Q40 for a large portion of the run. Q30 is way down here. You can see percent Q30 as a function of cycle. This small decreases as the run proceeds, as expected. The insert length for the 3 samples. So here are the 2 parents, and here's the proband, all nicely around 350 in this case, but we can tune that. So you can have shorter or longer depending on your application. And then the coverage is expected about half for the parents and 2x for the proband. So what do we see? And I'm sorry, this is a little bit small, but when we plug this into the interpretation engine of Genoox, the gene that immediately popped out is this one called CERKL, circle. And it was rare stop gain homozygous. And for those in the space, you can see that that's definitely one that would catch your attention. We saw a bunch of other promising things about it. It seemed to be predicted to be deleterious by a number of different engines. It was rare in Nomad, no homozygotes observed. So all of this built up our confidence that we should be looking into this more deeply. So we took a look in IGV. And you can see we had something like 72 supporting reads for the homozygote in the proband. So a very clear information there. And then both of the parents are carriers. Unfortunately, they both passed the broken copy to the child. We then check in ClinVar. It was classified previously as pathogenic by many groups. So again, our confidence continues to build. Does it correlate with our phenotype, which is early onset blindness? Genoox has a nice link out to the literature. And you can see right here, cone rod dystrophy, retinitis pigmentosa. So it was a very good fit to the phenotype. We return this to our collaborators. They need to do clinical sequencing to confirm, but they are very, very confident that this is actually the correct finding. So I'll conclude here and then take more questions. The message that I hope you were able to take from this is that the AVITI system enabled this benchtop study of an entire trio for $1,680 in reagent costs. The Elevate library prep was used. It's a simple, effective prep for whole genome sequencing and other applications. We were able to leverage this wonderful ecosystem with Google DeepTrio and Genoox for interpretation. And all of these components can easily be brought together. As I mentioned before, you can swap out some of these components for other components of your choice. We are very broadly compatible with other tools. As I